Cell death refers to a phenomenon occurring when cell damage is irreversible. Cell death may be very chaotic or very organized. Let's talk about necrosis, the chaotic death often described as dirty. Causing cell death after an external trauma, necrosis isn't programmed. For this reason, it is mostly associated with an inflammatory event, that is to say memory rupture, protein denaturation, and obviously strong inflammatory infiltration. From a morphological point of view, necrosis is extremely peculiar. From a histological point of view, cytoplasmatic RNA loss and protein denaturation cause cytoplasmatic eosinophil increase, also detectable in the tissue as a whole. Due to the loss in glycogen, the cytoplasm will seem vitreous and full of dense carbs due to molecular deterioration. Moreover, myelin figures depending on lipid peroxidation can be observed. From an ultrastructural point of view, the membrane will have numerous ruptures and mitochondria will be swollen. Nuclear modifications are even more peculiar. First, the nucleus gets smaller, pinoctic and extremely basophil, and then it is fragmented during a phenomenon called kyrexis. During kyrlysis, the nucleus is finally completely disintegrated. Because of its messy nature, this rupture leads to nuclei of different dimensions. There are different types of necrosis. The most peculiar are coagulative, co-liquidative, gangrenous, and caseous necrosis, while Stator Necrosis and Fibrino Necrosis are rarer. So-called for the architecture of its tissues, coagulative necrosis occurs after ischemia from vascular occlusion. In fact, despite damage leads to cell functional plaque and death, collagenous fibers are maintained anyway. For some days, the fragment of tissue with necrosis maintains its architecture, but over the time, necrotic tissue removal causes the loss of its architecture. Tissue digestion takes place afterwards through lysosomes and leukocytes, which are attracted by the inflammatory signals of cell death. With co-liquidative necrosis, the initial insult leads to a lesion of the tissue. The lowering of intracellular pH, the activation of lysosomes, and their hydrolysis is usually to such an extent that the tissue melts and collagenous fibers are destroyed. This type of cell death is typical of bacterial and fungal infections that are caused by microorganisms that can damage tissue through specific enzymes. Another example is that of cerebral infarction, commonly known as stroke. Stroke occurs because neurons are tied to glia. Since there is no fibers supporting the tissue, coagulative necrosis cannot occur. Cell death leads therefore to the loss of cell architecture. During co-liquidative necrosis, cells detaching from each other and creating abscesses may be observed under the microscope. In this case, necrosis is limited by the fibrous tissue. Nonetheless, cell debris of original tissue and neutrophils attracted by the inflammatory signals can be found at the site of necrosis. Dying due to coagulative necrosis, neutrophils form the so-called pus. Another form of coagulative necrosis is gangrenous necrosis. It affects different tissues, mostly those of the lower limbs. A common example of gangrenous necrosis is that of the diabetic foot. In this case, coagulative necrosis develops in ischemic tissues. When overinfected, the tissue liquefies and we can talk about wet gangrene. Caseous necrosis is typical of granulomatous infections, among which tuberculosis. Macrophages accumulate around the infected area to limit the infection and then create a granuloma. The enzymes generated by the macrophages and the lack of exchange with the outside lead to the degradation of the tissue, which becomes cheese-like and whitish, hence the name of this form of necrosis. A more peculiar example of necrosis is that of Stator Necrosis, typical of acute pancreatitis. In this infection, the release of pancreatic lipase causes the rupture of the adipocytes located in the retroperitoneum, from which triglycerides are released. The triglycerides are broken by the lipase that releases fatty acids combining with the calcium leaked out from dead cells. As a consequence, a process of saponification takes place. As you can see, they tend to form whitish areas similar to accumulation, mostly made up of esterified fatty acids. Fibrino necrosis is an even more peculiar case of necrosis that can be observed in many autoimmune pathologies. It is an immune complex perivascular deposition, that is to say, immunoglobulins binding antigens and complexed with fragments of complement. Immunocomplexes anchor the walls of small vessels, especially in limbs and kidney, triggering an inflammatory response with intense neutrophil exudation, which causes vessel damage and fibrin release through the damaged vessel. The fibrin release then forms an eosinophil ring around the vascular wall. Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.